Hello everyone, Mrs. Hanson here, and welcome to your Chapter 20 lessons. In this outline, we'll be focusing on carbohydrates. So let's take a peek at our chapter ahead of us. We'll introduce carbohydrates and talk about them as simple sugars, or disaccharides, or even polysaccharides. When we look at monosaccharides, those basic building blocks of larger structures, we'll look at Fischer formations as well as Haworth projections. Those are the cyclic forms of monosaccharides. We'll look at oxidation and reduction of monosaccharides, recalling that aldehydes and ketones can undergo oxidation and reduction. And we'll compare disaccharides to polysaccharides along the way as well. You do have some assigned reading in our chapters, focusing on the human body, such as useful carbohydrate derivatives and blood typing, along with some other areas as well. And they're focused out in your note pack for you. So here are our learning objectives. We'd like to identify the three major types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. We'll recognize the major structural features of those monosaccharides, the basic building blocks. We'll draw cyclic forms of monosaccharides and look at those as alpha or beta anomers. Do you hear that word? Anomers. The chemistry will involve oxidation and reduction reactions of those monosaccharides. We'll recognize the major structural features of disaccharides. The polysaccharides we'll look at are cellulose, starch, and glycogen. You'll give me examples of carbohydrates derivatives that contain amino acids, some amides, and carboxylate anions. And you'll be able to describe the role that carbohydrates played in determining your blood type. Are you type A, B, AB, or O? And of course, the positive versus negative part of that component as well. And that will be how we round out our chapter on carbohydrates. So thinking about carbohydrates in our new chapter, they are the largest group of organic molecules in nature. They comprise over 50% of the Earth's biomass. That's an extremely important molecule. They can be very simple or very complex, having as few as 3 or as many as thousands of carbons in their molecular structure. The glucose molecule, probably the one we're most familiar with, C6H12O6, it's metabolized for energy in our cells. The sucrose of table sugar, which is a disaccharide, and the cellulose of plants, well, those are polysaccharides. Those plant stems and tree trunks are all examples of carbohydrates. So these carbohydrates, or sugars as we commonly call them, are found on cell surfaces, and what type of sugar present or absence will determine your blood type. They form the backbone of DNA molecules. They carry all the genetic information in the cell. And unlike lipids from our last chapter, which were composed mainly of carbon-to-carbon and carbon-to-hydrogen bonds, very few functional groups, remember they were the very boring compounds that were nonpolar, carbohydrates have many, many polar functional groups whose structures and properties are understood by really just applying the basic principles of organic chemistry. So let's dive in and begin. Section 1, carbohydrates. They're called sugars and starches and are polyhydroxyaldehydes or ketones. Poly, of course, meaning many. So when I hear polyhydroxyl, aldehydes and ketones, those are the major functional groups found in our simple carbohydrates called monosaccharides. Let's investigate very closely two common monosaccharides. The first we'll draw is a glucose molecule. Notice that a glucose molecule has an aldehyde functional group at carbon number 1. The CHO, if you recall, has a carbonyl carbon, C double bond O, and it's attached to a hydrogen as its fourth bond. And it's leading down, of course, to a next carbon. So notice that you have here the aldehyde at carbon number 1. At carbon 2, we have an alcohol. At carbon 3, another alcohol. At carbon 4, another alcohol. Carbon 5, yet another alcohol. And again at carbon 6, the last alcohol. They are polyhydroxy, meaning many alcohol groups. And glucose happens to have at carbon number 1 contains an aldehyde. Now let's compare that, if you will, to the second monosaccharide shown to us on this page, and it's called fructose, fruit sugar. Glucose commonly found in blood, fructose, another simple sugar. Now notice here at carbon number 1, let's make this purple, carbon 1, 2, 3, 4, 5, 6. It's at carbon 2 that we see our carbonyl group. So now we know to classify this as a ketone. Carbon 2 contains the ketone functional group. Now that means we still have all kinds of alcohols as well. Carbon 1 contains an alcohol, so does 3, 4, 5, and carbon 6. They are polyhydroxy aldehydes, as in the case of glucose, or ketones, as in the case of fructose. These are two very common monosaccharides. When we draw these into our notes, we're drawing glucose as an aldehyde and fructose as a ketone, highlighting that they are polyhydroxy aldehydes or ketones. Disaccharides are composed of two monosaccharides joined together. And you can see a typical formula would be C12H22O11, whereas the simple sugar monosaccharide was C6H12O6. When we hook together two monosaccharides, to join them, we have to remove a water, and we'll do that a little bit later in our notes. Here is a very common disaccharide called lactose. Lactose is the milk sugar, and we use an enzyme called lactase to digest this sugar. People who are lactose intolerant lack the enzyme lactase. We know that the ASE ending indicates the name of an enzyme. Now to draw this, we are going to draw a six-membered ring where at the top right corner, you're going to place an oxygen of a six-membered ring. So draw with me, and I'll draw it with you if you'd like. When we take a six-membered ring, make the first top right the oxygen, and then we're going to just simply draw the rest of a six-membered ring as units of carbon. Alrighty. Now, when we think about how we're looking at lactose as a six-membered ring, carbon number 1 is the site of attachment. Alrighty, so we're going to learn to label this as the acetyl carbon. We'll come back to that. Let's put our OH at carbon number 2 going down. At carbon number 3, the OH points up. At carbon number 4, the OH also points up. And at carbon number 5, it leads up to another carbon. So here you have CH2OH up here as carbon number 6. Let me renumber this so you can see it. Carbon 1, 2, 3, 4, 5, and carbon 6 in this monomer is pointing up. Now down here, the H leads up here to an O, and that begins the next point of attachment. So this is not on the oxygen, it's attached to a carbon attached to here, so make sure we do this correct. Let me just clean that part up where carbon 1 is. This is the anomeric carbon. So here is an H leading up to an O leading up to another H, and right here is the carbon that's attached. Now, again, draw a six-membered ring where the top right is the oxygen, and then you come over and down to form the linkage. That's as best as I can do. I hope yours is looking as good as mine. Right here, we have the carbon on the second monomer. And this right here, just to keep numbering for us, we have carbon 1, 2, 3, 4. Here's carbon 5. And then notice that we climb up to carbon 6. CH2OH, this would be carbon 6. At carbon number 1, the OH is pointing down. At carbon 2, the OH is pointing down. At carbon 3, the OH is pointing up. Here's carbon 4, its OH pointing there, and then up there for carbon 6. Now, find with me where you find the acetyl carbon. Now let's remember what that means from a previous chapter. You want to find a carbon that's attached to two oxygens. Notice this carbon is attached to two oxygens that's attached to another carbon. So here's a carbon attached, and yet here's another carbon that's attached. So we have an acetyl carbon. The acetyl carbon is the carbon that's attached to two oxygens that both lead on to other carbons. The acetyl carbon is carbon 1 in this first sugar. Remember how we labeled those around? Remember that a monomer generally has the formula C6H12O6. In a six-membered ring, however, one of the members is actually an oxygen, making the sixth member up high off of carbon 5. And we're going to practice drawing these cyclic structures very soon. We have labeled, in this particular model, we've drawn lactose, and so far it highlighted in blue, I've labeled the acetyl carbon. Now let's find the hemiacetyl, and I'm going to highlight that in pink right here. Now remember what a hemiacetyl is. It's a carbon attached to two oxygens, but only one side leads to another carbon and the other side leads to a hydroxyl group, an OH. So the hemiacetyl is now highlighted in pink. So I'm going to just make sure we see when I highlight pink, that's the hemiacetyl. And when I highlighted with blue, that was the acetyl carbon. Then we're asked to go through and find, are the alcohols primary, secondary, or tertiary in our disaccharide called lactose? I remember that if a carbon attached to the oxygen is attached to two other carbons, we'll label that as a secondary alcohol, such as this right here. Here's a carbon attached to the alcohol, attached to two other carbons. This is a secondary alcohol. As is this at carbon 3. This carbon is attached to two other carbons, so this is a secondary alcohol. As is carbon 4. Carbons attached to two other carbons. How about up here at carbon 6? Well, this alcohol is attached to just one carbon, so at carbon 6 we have a primary alcohol. And of course here, this carbon here is not an alcohol, it's an acetyl. Up here we had primary, this guy here is secondary, secondary, secondary alcohols. There are no tertiary alcohols in our disaccharide compound. We've just practiced drawing a disaccharide called lactose. Disaccharides are composed of two monosaccharides, two cyclic structures where we have six-membered rings, where the position of the oxygen here is the top right corner of a six-membered ring, meaning that the sixth carbon is outside the ring. All right, good work. We've talked about monosaccharides. disaccharides, and the third category of carbohydrates are called polysaccharides, and they're composed of at least 3 or more monosaccharide units joined together. So let's draw as an example starch. Starch here, this is formed from bread. One from starch is a polysaccharide. And again, we're going to draw a 6-membered ring where the top right corner of our 6-membered ring is an oxygen group. And then just simply draw the ring. At position number 1 is to the right of the ring, to the right of the oxygen. Position number 2, 3, 4, 5, and that means from carbon 5 pointing up is where we find carbon 6. To the right of the oxygen is carbon 1. And right here will be our linkage to the next monosaccharide. So let's kind of show the linkage is going, let me get back to red. They're pointing down to the oxygen and then back up. And then I draw a next membered ring, 6-membered ring where the top right corner is an oxygen. I point down and then back up and I'll join to the next ring. 1, 2, 3, 4, 5. Up here, however, remember this is the oxygen when I come back up, and I can just keep drawing forever and ever. This is a very long molecule, a 6-membered ring where the top right is always an oxygen. And then you just kind of do this to say that continues on and on and on, and you can say that over here, it continues on and on and on. Now that we have them linked together, let's remember the anomeric carbon is at carbon 1. At carbon number 2, the OH is pointing down. So let's make sure we see the OH pointing down at carbon 2 all the way through. At carbon number 3, we see the OH pointing up. So we'll do that all the way through. At carbon 4 is a hydrogen up, and so is at carbon 1. Now what happens at carbon 5? We had said that that's where it leads to your next carbon, giving us 6 carbons in our cyclic structure. So that CH2OH is the 6th carbon. There, I just drew 4 repeating units just as the slide shows, and we're getting a feel of what these polysaccharides will look like. What we're looking at is a acetyl carbon, which is carbon number 1 in each one of these, and I can highlight that so you can clearly see in each case here. Here's an anomeric right here. The acetyl carbon is labeled with a little star. That anomeric carbon, we'll learn what that means. It's the linkage between one monosaccharide to the next. But it's an acetyl where I've highlighted in yellow. Acetyl carbons are starred in this original picture and now highlighted in yellow. And they're always carbon 1. It is directly right to the oxygen in that 6-membered ring. So carbohydrates, they can be monosaccharides, which are basic building blocks, disaccharides, which are joined by 2 monomers, and polysaccharides, which are many, many, many monomers long. Carbohydrates are synthesized in green plants through the process of photosynthesis. So photosynthesis, of course, produces glucose, whereby energy from the sun is stored as chemical energy in the bonds of these sugars. And of course, when we need bursts of energy, like say during exercise or even just cellular metabolism, we break down the glucose to release the stored energy found in the chemical bonds. So in the forward direction, carbohydrate, 6 members of carbon dioxide, 6 CO2s, combines with 6 water molecules in the presence of sunlight and chlorophyll to create glucose molecule, C6H12O6, and releasing oxygen along the way. So in the forward direction, we call this photosynthesis. This is building glucose. And this requires energy. It only occurs in the presence of sunlight and the chlorophyll in green plants for the site of reaction. Of course, the photosynthesis then would be the endothermic process. Heat absorbing to store the larger molecule. The reverse reaction is metabolism. Sometimes you hear this called as cellular respiration. And that's a breaking down the glucose to release the energy. So cellular respiration or metabolism breaks down glucose, and photosynthesis builds glucose. They are indeed reverse reactions of one another. Monosaccharides are the simplest carbohydrates. They can have anywhere from 3 carbons to 6 carbons in their structure. Typically, they are drawn as a vertical chain, a vertical structure. Generally vertical in a chain with an aldehyde and ketone and many, many hydroxyl groups. So at carbon 1, if we have the carbonyl group at carbon 1, we know that would be an aldehyde. We're going to refer to that as aldose. If the carbonyl group is at carbon 2, then we have a ketone, and we'll refer to that as a ketose. Aldose stands for aldehyde sugars, and ketose stands for ketone sugars. Anywhere from typically 3 to 6 carbons long, we have a monosaccharide drawn in a vertical fashion. Aldehyde sugars are called aldoses. Ketone sugars are called ketoses, anywhere from 3 to 6 carbons long. Well, let's take a look at some simple aldose and ketose structures. The simplest aldose is called glyceraldehyde. You say that with me, glyceraldehyde. And the simplest ketose is called dihydroxyacetone. Dihydroxyacetone. They are indeed constitutional isomers of one another, sharing the same molecular formula, C3H6O3, but they are bonded in a completely different order. Let's take a look at the simplest aldose. The sugar with an aldehyde is known as an aldose. A 3-carbon unit, 1, 2, 3, where at carbon 1 is the aldehyde position, so it's called an aldose. Carbon 2 contains a hydroxy group, and carbon 3 also contains another hydroxyl group, polyhydroxyl aldehyde. Now, when we look at the most simplest ketone, a sugar that is a ketone structure is known as a ketose. We place the carbonyl carbon at carbon 2. So that means at carbon 2 we have a ketone, and at carbon 1 and 3 we find the alcohol. We've just drawn the simplest structure, those constitutional isomers of each other, called glyceraldehyde, a 3-carbon aldehyde sugar, and dihydroxyacetone, which is a ketone sugar, so we call it a ketose. These monosaccharides are characterized by the number of carbon atoms in its chain. We just drew triose. Both of them had 3 carbons. If they had 4 carbons, you'd hear them called a tetrose. If they had 5 carbons, they would be called a pentose. And 6 carbons would be called a hexose. When I hear OSE, think sugars. So a sugar monomer, a monosaccharide containing 3 carbons, is a triose, tetrose, pentose, and hexose. Now, those terms are commonly combined with the words aldose if it's an aldehyde, or ketose if it's a ketone. A moment ago, we just drew glyceraldehyde. It had 3 carbons, and the first carbon had the double bond O. So there was your aldehyde. So we would call that an aldotriose. Aldo because it's an aldehyde, triose because we know it has 3 carbons. We just drew a moment ago dihydroxy. So here were the OH groups at carbon 1 and 3, and the carbonyl group here was a ketone. Since it had 3 carbons in the structure and it was a ketone, dihydroxyacetone is known as a ketotriose. Fun words to say, aren't they? Glyceraldehyde, a 3-carbon unit, so triose, but I put the prefix aldo in front of it to let you know that it's an aldehyde. And a 3-carbon unit with a ketone would be a ketotriose. Shall we try some? Let's practice. Where it tells us to classify each monosaccharide by the type of carbonyl group and the number of carbons in the chain. So I first want to recognize where we find the carbonyl group. And I'll just highlight those so we can bring our attention to those as we solve those problems. So when I recognize where they are, ribose, for example, will be an aldose. Letter B would be a ketose. Down here, an aldehyde is known as an aldose, and ketones are known as ketose. So I just wanted to do step 1, what type of carbonyl group we have. Classify each by the type of carbonyl group. Okay, so that's done. But now what about the number of the carbons? So in this structure we have 1, 2, 3, 4, 5 carbons in the sugar called ribose. Maybe you remember that sugar from ribonucleic acid in an RNA structure. So we said a moment ago when we said a 5 sugar, remember how we said those are called pentose? And we're just going to put the prefix aldo in front of it to let you know it's an aldehyde structure. An aldopentose. Here we have in letter B 6 carbons in that chain for the monomer called fructose, fruit sugar, and we said it was a ketose, so we put that prefix in front. Ketohexose is how we'll say that name for representing what type of carbonyl group as well as the number of carbons in the chain. How about down below? 1, 2, 3, 4, 5 carbons in this structure as well. What makes this structure of ribose different than arabinose is the location of the side on the hydroxyl groups. Do you notice that at carbon 2 in ribose, we have the hydroxyl group pointing to the right? But here in arabinose, we would have OH pointing to the left. So therefore, these are diastereomers of one another, aren't they? Diastereomers, they're not mirror images. We said this was an aldo, whoops, that's supposed to be an A. Aldopentose. Here we have 1, 2, 3, 4 carbons in that chain. So we'll use the prefix aldo to tell you it's an aldehyde, and then tetrose for 4 carbons. And 1, 2, 3, 4 carbons here again, but the prefix will be ketotetrose. Tetra is 4, tetrose, giving us the sugar compound. How'd you do with those? Did you write aldopentose, ketohexose, aldopentose, aldotetrose, and ketotetrose for those particular structures? We've practiced recognizing if it's an aldehyde or ketone by using the prefix aldo or keto, and then just simply counting the carbons in the chain to tell us how many carbons are in the monomer. Let's draw the structure of an aldotetrose. Well, when I think about aldotetrose, that's telling me I've got 4 carbons in a chain, and the prefix, so that's tetros, meaning 4, and that at carbon 1 is going to be an aldehyde. So you can show that by saying CHO, but remember that's really telling you that it's an aldehyde functional group. So just kind of know CHO, that's a carbonyl carbon there. Leading down to carbon 2, and I absolutely can put these OHs on either side. It doesn't matter. I can draw any type of enantiomer of the structure. And then we'll terminate down here, of course, with another hydroxyl group. So an aldehyde, looking at carbon 1 is the aldehyde. Carbon 2, 3, and 4 all contain alcohol groups. How about trying the next 2? What would a ketopentose look like? And an aldohexose. Draw those and come back to the video when you're ready to check. All right, welcome back. I can see that we have a 5 carbon chain, 1, 2, 3, 4, 5, and at carbon number 2 is where the carbonyl group will be because it's a ketopentose, meaning we have alcohol groups on every other carbon. So at carbon 1 and at carbon 6, carbon 1, 2, 3, 4, 5. At carbon 1 and at carbon 5, we terminate with alcohols, and carbon 3 and 4 create chiral centers, and you can put the OH on either side. They're both absolutely correct. So that's one example of a ketopentose. And how about an aldohexose? Well, that would be drawing 6 carbons, 1, 2, 3, 4, 5, 6. I know that at carbon 1 would be the carbonyl carbon because it's an aldehyde. And therefore, all of the other chiral carbons can be on either side. I can do OHs all on one side, or I can mix and mingle. It doesn't matter. They're all valid structures, just so that you have an H and OH on each chiral carbon. So there are lots of opportunities for how you decide to put OH and H on each of those carbons to come up with all kinds of diastereomers. We're drawing aldohexose, 6 carbons with an aldehyde at carbon 1. Ketopentose is 5 carbons with a ketone at carbon 2. And then an aldotetrose, 4 carbons with the aldehyde at carbon 1. Every other carbon has a hydroxyl group on it, and we weren't told any difference. You could just pick what side to place it on, the left or right side of the chiral carbon. And here are some examples. These may or may not match mine. It looks like I just mixed and mingles for whatever hydroxyl groups, and you may have as well. So we have more than one correct structure for each of these examples. We know that some physical properties of these monosaccharides when we think about sugars. So one of the very first things you think about with sugars is how sweet they taste. Monosaccharides are very sweet tasting, but they vary greatly with how sweet they are. We know that they're polar compounds and they have very high melting points. And the presence of just so many polar functional groups, all capable of hydrogen bonding, makes sugars very, very soluble in water. So 3 common traits, 3 physical qualities of sugars. They're very sweet tasting, very polar compounds capable of hydrogen bonding, making them have high intermolecular attractions and thus high melting points and very water soluble. So if we think that through, let's rank the following compounds in order of increasing water solubility. We would have glucose, and we learned that glucose is a polyhydroxy aldehyde. If you just think about what I'm saying, it has 6 carbons, right? And then here, of course, you'd have a CHO, and then all of these would contain hydroxyl groups. Polyhydroxyl aldehyde, right? So or it could be a ketone. Remember, just saying they can be either one for these monosaccharides. The next one we have is just boring hexane, right? 1, 2, 3, 4, 5, 6. This is called hexane. It's C6H14 is its overall molecular formula. And then in the middle we have 1-decanol. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then an OH group coming off as an alcohol. The least soluble in water is, of course, the hydrocarbon straight chain, no polarity. Kind of a boring molecule, isn't it? Likes dissolve likes, so if it's nonpolar, it is not going to be very water soluble. The alcohol group as 1-decanol has a very large nonpolar chain. It has a little polarity here at the end, so that might be, it would be highly unlikely to be soluble with such a long tail. But of course in glucose it would be highly soluble due to all of those alcohol chains, meaning all of these have potential here to set up hydrogen bonding with a nice water molecule which also has hydrogen bonding. So the least soluble hexane, followed by the alcohol, and the most soluble, of course, would be your sugar, the glucose. When we think about monosaccharides and all of those chiral centers, the easiest way to draw these is by using what we've learned as a Fischer projection. All of the carbohydrates, well, I say all except for the ketone, the dihydroxy ketone, this has no chiral center. That's what we learned as the simplest ketose. Dihydroxyacetone has no chiral center, but it really is the only sugar that does not have at least one chiral center. And we know that the most, easiest way, the most familiar way for us of writing out these structures is using what's called a Fischer projection. So for example, if we were to redraw our glyceraldehyde, which was our simplest aldehyde sugar, using dashes and wedges, we see that there is one chiral carbon right here at chiral carbon at 2. In a 3-carbon chain, carbon number 2 is a chiral center. And remember that Fischer projections on the vertical lines, those are the ones that go up and down, those are dashes. And on the horizontal lines, those are coming out to hug you from the page, those are wedges. When we see that the chiral center is at carbon 2, we can draw its enantiomer, its mirror image, and come up with the enantiomer known as L-glyceraldehyde. The first occurs naturally as the D position, and notice that it's on the right side. The hydroxyl group is on the right side for the D position. And in an unnatural isomer, we see that the hydroxyl group is on the left side. This is known as the L-glyceraldehyde. The prefix D is used to tell us that the hydroxyl is on the right side of the chiral carbon, and the prefix L is used to tell us that the hydroxyl group is on the left side of the chiral carbon. Now, if you were to draw these again using Fischer projections, remember that we would simply use a cross to represent the chiral carbon. At the top, we would place our aldehyde, CHO, and at the bottom, we would place the third carbon, CH2OH. On the right side is the OH, and on the left side is the H. This is the D configuration. D actually stands for dextro-rotary, spinning to the right. If I redraw the L configuration, the only difference now in its mirror image is the hydroxyl group is over here pointing to the left side, making it the L configuration, which stands for levorotary, meaning left. In nature, we favor the D configuration. In nature, you will not find an L enantiomer of these sugars. I'm just reporting what we find in nature. For whatever reason, the Ds are dominant. So we draw these Fischer projections and come up with the enantiomers of glyceraldehyde. And that's just holding up the mirror and drawing, really just converting everything that's on the horizontal lines, flipping their sides. The unnatural isomer, of course, is the L configuration. On the next page there, the prefix D is used to tell us it's on the right side. The prefix L is when it's on the left side. D relates to the hydroxyl group. If it's on the right side, call it a D configuration. And if it's on the L, left side, that's the hydroxyl group on the corner there. And of course, we just know to turn these dashes and wedges into straight lines where the chiral carbons are across. Do not draw the carbons there, but it's at the cross of both lines, the intersection. That's the chiral carbon. So glucose has 4 chirality centers as it's drawn. In your note pack there, you can see that we have the chiral carbon line structure of glucose. And I can count with you the 4 chiral centers. 1, 2, 3, 4. They're designated with little stars on the original picture. Remember that glucose contains 6 sugars, or 6 carbons in the sugar. Carbon number 1 contains an aldehyde. So we would call this an aldohexose, a 6-carboned monomer with an aldehyde at carbon 1. Now, when you draw the Fischer projection, just replace each chirality center with a cross. So you'll just draw a straight line down. Remember, at carbon 1, we have the aldehyde. At carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6 is the last alcohol. It is not a chiral center, so we just write it out as CH2OH. Now keep in mind, it matters where the alcohol is in terms of giving us the correct structure for glucose. At carbon number 1, 2, 3, 4, 5, 6. Carbon number 3 has the hydroxyl group on the left side. At carbon 2, 4, and 5, the hydroxyl groups are all on the right side. So how do we determine if it's a D or an L configuration? And the trick there is to find the farthest chiral center away from the aldehyde. The farthest away will be used to determine if it's a D or an L configuration. At carbon 1, 2, 3, 4, 5. C5 is the farthest chiral carbon. And now what would be its configuration? It's on the right side, that's spinning dextro rotary. So this would be, in terms of its configuration, D-glucose, which we suspected because that's what's favored in nature. You really don't find an L configuration in nature of a glucose molecule. So when we're finding D and L, we find it by the farthest from the carbonyl group. If right down here is the last chiral carbon, if it's on the right side, it's the D configuration. If it's on the left side, it's the L configuration. Notice that this is the natural sugar and this is an unnatural sugar. All naturally occurring sugars are the D conformation. The L's are not found in nature. Alrighty, so just find the last chiral center. If it's on the right side, the hydroxyl group is on the right side, you name that as the D configuration, D-glucose. And if it's on the left side, you would name that L-glucose. And clearly you can see that this is the naturally occurring enantiomer, and an unnatural enantiomer would be the left side. All right, good repeating there. Let's give you a moment to process, think this through. Pausing the video as you process this. This is an aldopentose. Find all the chirality centers, classify it as D or L, and then draw its enantiomer. Pause the video and work ahead of me. All right, welcome back. Well, first we know it's an aldopentose because at carbon number 1, we can see that there's an aldehyde. So this is the aldo prefix. Each place that we see a cross, we recognize that as a chiral carbon. So we have 3 chiral carbons. Those are found at C2, C3, and C4. These are all chiral centers. To classify from a D or an L perspective, we need to go in and find the farthest chiral center from the alcohol or from the aldehyde. And that happens to be at carbon 4 in this situation. And we see that it's on the right side, as we suspected if this is a natural sugar. So this would be D-ribose. D stands for the right side, dextro rotary. Dextro is right. L, of course, would be the left side. It's levo-rotary. So this is the D isotope. To draw its enantiomer, we can simply redraw. Carbon 1 is the aldehyde. Carbon 5 is not chiral, so we just CH2OH. And then the chiral centers, we just simply flip the order. OH to the left this time. OH again to the left this time, and OH again to the left this time. So we're just redrawing what we see from a perspective of a mirror image. And of course, at this point, we drew L-ribose, which is the unnatural way to find the sugar. For each of these, label the chirality centers, tell me if it's D or L, and draw its enantiomer. Pause and work ahead. Come back when you're ready to check. All right, welcome back. Well, first we know that all chiral carbons are the centers of the intersections. So I can quickly find all the chiral carbons as being the cross between the vertical and the horizontal lines in our Fischer projections. To focus if it's a D or an L, we look at the very farthest chiral center and determine if the hydroxyl group is to the left or to the right. So with carbon 1, 2, 3, 4, this would be an aldotetrose. Carbon 3 has the hydroxyl group to the right. That makes this the D enantiomer. To draw its chiral center, its enantiomer, we had 2 chiral carbons. We just flip the order. So everything that's on the right side goes left and vice versa. And here is the L configuration. 1, 2, 3, 4, 5 carbons. So this is an aldopentose. The farthest carbon that's a chiral center is carbon number 4, and here the OH is on the left. So this enantiomer is an L configuration. So when I draw its mirror image, CHO, the aldehyde, CH2OH, oh, I need another line. So I'm just going to flip what I have on the left to the have on the right. So over here I would have HOH, OH, H, HOH. And of course now this is on the right side, so we have a D enantiomer. 1, 2, 3, 4, 5 carbons again. So this is also an aldopentose. The farthest chiral carbon places the hydroxyl group on the right. At C4, hydroxy is on the right. And so therefore we know it will be a D enantiomer. To draw its mirror image, we had 3 chiral carbons. Up at carbon 1 is your aldehyde. The last carbon is not chiral, so just CH2OH. And we just flip what's on the sides from the opposite. So HOH and then OHH. And now we've drawn the L enantiomer. I bet you're doing great. Very common monosaccharides, glucose, sometimes you hear it called dextrose as well. Glucose is known as blood sugar and the most abundant monosaccharide. We can see that the blood sugar in a normal concentration are found at typical values of 70 to 110, and that is a microgram per deciliter. Excessive glucose is stored. Did I say micro? This is milligram per deciliter, just so you can see that. Excess glucose, of course, is stored as a polysaccharide, and we'll get that towards the end of our chapter. We'll call that glycogen, which is stored fat. And then insulin in our body, of course, is used to regulate glucose levels, and you're going to read more about that in a health and reading assignment later on in this very chapter. Insulin, of course, regulates blood glucose by signaling the body to uptake excess levels of glucose and store them in the form of glycogen. It's patients with diabetes who cannot produce sufficient amounts of insulin to adequately regulate their blood sugar levels, and sometimes they have to take injections or wear pumps or so forth. Some of our classmates have this very condition right now. Here's some snapshots of other common monosaccharides. You can see that monosaccharides can be an aldehyde, such as the glucose molecule. Remember, the glucose molecule is an aldohexose. It has 6 carbons in its structure. At carbon number 1 is an aldehyde. You can see it's a D configuration because the farthest carbon from the aldehyde group is on the right side of our Fischer projection. D-glucose is an aldohexose. Carbon 3 places the alcohol on the left side. Carbon 2, 4, and 5 place it all on the right side. Here's a second D-galactose, a simple sugar. Again, this is a 6-carbon chain. Notice now what's happened at carbon number 4, just to kind of compare these two structures. Carbon 4 and glucose place the hydroxyl group on the right. Galactose places it on the left. That is just a stereoisomer, right? They're diastereomers. They're not mirror images. And fructose, well, it's a ketose altogether. So we have a different conformation here, that we have a 6-carbon chain, but we would call it a ketohexose because that carbon 2 is a ketone instead of the alcohol. So you've drawn in an aldohexose, looking at the position of the hydroxyl groups to compare D-glucose to D-galactose. And the next one you've drawn in is D-fructose, knowing that it is a ketohexose. Oh, your tongue gets tied saying all of these words. Common monosaccharides also include galactose, which is one of the components really in lactose, which is milk sugar. We drew that a moment ago and said lactose is a disaccharide. It's one of the 2 sugars. Patients with galactosemia, they lack that enzyme needed to metabolize galactose. And you see that accumulating, causing cataracts or even cirrhosis of the liver. And fructose, that 3rd sugar you just drew, is one of the common components of table sugar, that disaccharide known as sucrose. We found that it's a ketohexose found in honey. It's almost twice as sweet as sucrose, which is table sugar, with the same number of calories per gram. Many people like to sweeten things with honey since it's much more potent. I think we found a good place to pause here. We've done first 2 sections in our chapter. We'll take a little break, come back to the lessons when ready for lesson 2.